Hey everyone, and welcome. I am absolutely thrilled you're here. Today, on behalf of BioMitra, your friend in life sciences, we're diving into a brand new explainer on biological databases. Now, as biotech undergrads, I want you to think about a single microscopic cell for just a second. Got it? Okay, now think about the massive global engines of scientific discovery. How in the world do we get from one to the other? Well, today, we're tracing what I like to call the hero's journey of a data point. We are going to follow a tiny biological fragment as it transforms from a physical sample sitting in a lab into a literal computational powerhouse driving global research. Okay, let's dive right into this. Here is our roadmap for today. We'll be hitting what is biological data, sources of information, the need for databases, the database hierarchy, submitting and retrieving, and finally, we'll tie it all together with the complete life cycle. Starting right off with Section 1, what exactly is biological data? So let's start with a massive question. Every single day, laboratories all over the planet are extracting raw genetic code from cells. To put this scale into perspective for you, repositories like GenBank are practically doubling in size every 18 months. We are dealing with petabytes of data. But where does all that extracted code actually go? Well, to answer that, we first need to categorize what we're looking at, and it all starts right in the architecture of the cell itself. Deep in the nucleus, we have sequence data. That's your raw genetic code of DNA and RNA. Move outward a bit and we hit structure data, which maps the 3D atomic coordinates of those biomolecules. Then we have expression data, telling us the actual quantitative levels of gene transcription. We also capture interaction data, mapping out those incredibly complex networks of molecules binding and signaling to each other. And finally, zoomed all the way out, we have phenotype data, the observable traits and physiological outcomes of the organism. Moving right along to Section 2, sources of information. Now that we know what the data is, where does it all actually come from? Basically, it funnels in across three distinct pillars. On the far left, we have experimental data, affectionately known as the wet lab. This data comes directly from physical samples. Think sequencing machines or X-ray crystallography. It has extremely high authenticity, but, also high noise. In the middle is public data, or the global vault. These are existing datasets downloaded from open access repositories. They're pre-formatted, highly accessible, and ready for researchers anywhere. And finally, on the right, we have simulated data from the dry lab. This is purely theoretical data, generated in silico using computational models and algorithms, and it's mostly used for benchmarking our tools. Section 3, the need for databases. So we're generating all this incredible data across those three pillars, which leads us to a huge core conflict in modern biology, dealing with a chaotic web of unmanaged data silos. Just imagine fragmented, isolated text files scattered across completely unlinked computers all over the world with zero standard formatting. It is a nightmare for collaboration. But this brilliantly illustrates the contrast. We move from that chaos into the highly structured, perfectly interconnected ecosystem of biological databases. Suddenly, the flow of data is logical, it's accessible, and it's primed for massive computational analysis. To actually make that structured ecosystem work, these databases act as vital scientific infrastructure by nailing three core functions. First up is centralization. They connect global scientific knowledge into centralized hubs. Next is standardization. They enforce universal machine-readable formats, which means a computer in Tokyo can seamlessly read a file generated in London. And third is annotation. Honestly, this is arguably the most important one. Think of annotation like sticking highly detailed Post-it notes full of vital biological context onto the raw code. It ensures a random string of A, T, G, and C actually means something functional to a researcher. All right, on to Section 4, the database hierarchy. We know why we need them, but how do they actually function? Well, biological data gains significant value as it moves through a really strict filtration pipeline. Raw experimental sequences coming out of the lab are first dumped into a primary database. This is purely archival, but then they pass through a rigorous curation and annotation filter. This is where experts review the data, check it for errors, and attach all that critical biological meaning we just talked about. Once filtered, that data lands in a secondary database, which is highly structured and curated. Finally, it links into a composite database, which essentially integrates multiple distinct sources together. I absolutely love this analogy. It breaks it down so well. Think of a primary database like a massive public library archive. It's raw and curated experimental data submitted directly by researchers. And it has high redundancy because, hey, multiple labs might submit the exact same gene. Now, a secondary database is much more like an edited, peer-reviewed textbook. It is heavily curated by expert database teams, consolidated to have very low redundancy and packed with computational data. And a composite database? That's like your master search engine. Created by system integrators, it acts as a portal linking data from multiple sources without actually holding its own redundant archives. Section 5. Submitting and retrieving. Now that we understand the architecture, how do researchers actually push data into it? Let's compare two distinct sequence submission tools. First, Bankit. This is a wonderfully simple web-based tool. It requires absolutely no software installation, and it guides users through structured web forms. It's perfect for simple, single sequences. Contrast that with Sequin. This is a standalone software application you actually have to install on your computer. It allows for offline editing, complex validation, graphical viewing, the whole shebang. It is the heavy lifter, ideal for massive, complex submissions, like if you're uploading an entirely sequenced whole genome. Now, what's really interesting here is how we pull that data back out. Global retrieval topologies take some totally different approaches. Entree, which is the NCBI's integrated search system, is amazing at cross-linking records, but strictly within its own ecosystem. That means you can jump seamlessly from a PubMed scientific paper directly to a GenBank DNA structure. On the flip side, the Sequence Retrieval System, or SRS, casts a much wider, truly global net. It's an indexing system that links completely disparate biological databases all around the world, letting you run complex queries across formats way outside of just one organization's walled garden. Okay, so you retrieve the data, but you have to actually be able to read it. Welcome to the Rosetta Stone of file formats. Think of the FASTA format like a simple, plain envelope. It's a universal, plain text format prefixed by a single greater than sign. It's highly streamlined, stripped of any extra text, and designed specifically for super fast computational alignment. GenBank, however, is more like a heavily detailed clipboard. It is incredibly rich, heavily annotated, and human-readable. It contains extensive biological context using fielded metadata, things like the sequence's locus, definition, and accession numbers. But what happens when you retrieve a rich GenBank file, but the specific alignment software you're using only accepts that streamlined FASTA format? Boom. You've just hit the interoperability problem. This is where digital conversion tools like ReadSeek step in as translators. You just feed the rich GenBank data into the conversion tool, it extracts the core biological data from the schema, and instantly restructures it into a FASTA format. This ensures seamless data flow through your analytical pipeline so the process never breaks down, and you never have to waste time doing manual re-entry. Bringing it all together in Section 6, the complete lifecycle. We've looked at all the individual pieces today, the types, the sources, the databases, the tools. Now let's map out the exact roadmap. It's a clean 5-step biological data lifecycle. Step 1, generate, identifying the types and sources of your biological data. Step 2, store, understanding how it lives in primary, secondary, or composite databases. Step 3, submit, physically pushing sequences using tools like Sequin or Bankit. Step 4, retrieve, querying systems globally via Entree or SRS. And finally, Step 5, process, utilizing those formats and conversion tools to get the data completely ready for your final analysis. So the crucial point is really seeing how this complete ecosystem blueprint fits together in practice. Let's trace it one last time. In Step 1, a physical organism in the wet lab yields a sample that becomes raw experimental DNA. In Step 2, a researcher uploads that raw data using the Bankit web tool. Step 3, that sequence gets safely archived in the global vault of a primary database like GenBank. Over to Step 4, expert curators verify and annotate that gene, elevating it up to a secondary database like RefSeq. And finally, in Steps 5 and 6, completely different scientists on the exact opposite side of the world use Entry to search for it, download the FASTA file, and process it for their own computational analysis. This proves that biological databases are not just dusty old archives, they are active, deeply interconnected ecosystems that drive global scientific discovery literally every single day. Thanks so much for joining me on this explainer today with BioMitra. Before we wrap up, I want to end on a bit of a provocative thought for you to carry into your undergrad studies. If just one properly managed organism's DNA can fuel this entire globally interconnected ecosystem, leading to massive leaps in genomics and artificial intelligence, what world-changing scientific discoveries are hiding right now in the data that you will manage tomorrow? Keep exploring, stay curious, and I'll catch you in the next explainer.